For more than 2,000 years, nobody had ever actually held a piece of confirmed ancient Israelite DNA. Every study of Jewish genetic history, and there have been many, worked backward, starting with modern populations scattered across Europe, North Africa, and the Middle East, and trying to infer what their ancestors 2,000 or 3,000 years ago might have looked like genetically. Nobody had a direct reference point. Nobody had the actual DNA of a person who had lived and died inside the biblical kingdoms of Israel or Judah, against which every later theory could finally be tested rather than guessed at. Every research team working on this question, no matter how sophisticated their tools, was essentially trying to reconstruct the shape of a source by studying only the ripples it left behind, centuries and continents away. The genetic equivalent of trying to describe a stone by watching where the water moved after it had already sunk out of sight. Then, in a small burial cave on a hillside near Jerusalem, that changed. The cave sits close to the modern town of Abu Ghosh, near a site called Kiryat Ye'arim, a name that appears in the Hebrew Bible itself, associated with the resting place of the Ark of the Covenant before it was brought to Jerusalem. Archaeologists led by Israel Finkelstein had been excavating the area for years, and inside this particular cave they found a family tomb dating to what scholars call the Iron II period, roughly 750 to 650 BCE, squarely within the era of the First Temple, when the kingdom of Judah still stood and Jerusalem was its capital. The tomb had been looted at some point in antiquity, its contents disturbed, the pottery scattered, valuable items long since carried off by whoever broke in centuries or millennia ago. But enough remained for a full excavation to recover a meaningful sample of that pottery and, crucially, human remains that had survived well enough to attempt something that had never successfully been done before. In 2023, a team that included the geneticist David Reich began the painstaking process of trying to extract ancient DNA directly from that Iron Age burial, targeting the petrous bone, a dense structure at the base of the skull, long known among paleogeneticists for preserving genetic material far better than almost any other part of the human skeleton, precisely because its density protects the DNA inside from much of the bacterial decay and environmental degradation that destroys genetic material elsewhere in a skeleton far more quickly. It worked. Researchers recovered usable genetic material from two individuals in the tomb, one male and one female, both of whom had lived and died during the era of the First Temple. This was, by any reasonable definition, the first time science had ever obtained confirmed ancient DNA from people who could be called ancient Israelites or Judahites in the most direct sense, buried in Judah's own territory during the biblical kingdom's own lifetime, rather than reconstructed centuries later from the DNA of their scattered descendants. To understand why this moment mattered so much and why it took so long to arrive, it helps to understand just how much time and how much upheaval separates the modern Jewish world from the ancient one this cave belonged to, and how many distinct historical ruptures sit between the two. The kingdoms of Israel and Judah existed as distinct biblical states through the Iron Age, Israel to the north, Judah to the south, with Jerusalem as its capital, until Israel fell to the Assyrian Empire in the 8th century BCE, and Judah fell to Babylon in the 6th, an event that brought the destruction of the First Temple and forced exile for a significant portion of Judah's population to Babylon itself, a rupture ancient texts describe as one of the defining traumas of the entire biblical narrative. Decades later, following Persia's conquest of Babylon, a portion of that exiled population returned and eventually rebuilt a second temple in Jerusalem, restoring a form of Jewish political and religious life in the region that continued through periods of Persian, Greek, and eventually Roman rule for roughly 6 centuries. That Second Temple period ended in 70 CE, when Roman forces destroyed Jerusalem and its temple following a Jewish revolt, an event that scattered Jewish communities across the Mediterranean world and beyond in a dispersal that would continue in various waves for the better part of 2,000 years. Communities formed and reformed across Europe, North Africa, the Middle East, and eventually far beyond, developing distinct languages, customs, and, over enough generations, distinct genetic profiles shaped by whichever populations they lived alongside and occasionally intermarried with. By the time modern genetics existed as a scientific tool at all, the Jewish world had already been geographically scattered for the better part of 2 millennia, and any study of Jewish ancestry was, by necessity, a study of that scattering rather than of the source it scattered from. This gap between the ancient source population and its modern dispersed descendants is exactly what fueled decades of genuine scholarly debate over Jewish genetic origins, debate that, without any actual ancient Israelite DNA to test against, had no way of being fully settled. Different researchers working from different modern population samples and different genetic markers reached different conclusions, and each new study seemed to add another layer to an already complicated picture rather than resolving it outright. Some studies emphasized shared Middle Eastern paternal lineages, linking Jewish communities across Europe, North Africa, and the Middle East to each other and to non-Jewish Levantine populations. Evidence generally interpreted as supporting a shared ancient origin in the region of ancient Israel and Judah, with the same broad Y chromosome lineages showing up again and again across Jewish communities that had otherwise been geographically separated for well over 1,000 years. Other studies focused specifically on maternal lineages within Ashkenazi Jewish communities, found that a majority of Ashkenazi mitochondrial DNA traced back to prehistoric European women rather than to the Near East, suggesting that conversion and intermarriage played a significant role in the formation of specific communities at specific points along the way, particularly in southern Europe, roughly 2,000 years ago, and later in West and Central Europe, a finding that on its surface seemed to sit in tension with the paternal evidence pointing toward the Middle East, until researchers began to understand both patterns as different, equally real threads within the same complicated migration story, rather than as contradictory claims. A separate hypothesis, popularized well outside strictly academic circles and often called the Khazar theory, proposed that a substantial share of Ashkenazi ancestry might trace back to the Khazars, a medieval Turkic-speaking polity north of the Caucasus whose ruling elite is historically documented to have adopted Judaism at some point in the 9th or 10th century. For decades, this idea circulated widely enough in both serious academic discussion and more popular writing that it became one of the most commonly repeated alternative explanations for Ashkenazi origins, sometimes treated as a serious contender and sometimes invoked for reasons that had less to do with genuine historical curiosity and more to do with broader arguments about identity and belonging. That specific idea has been examined directly using modern genetic tools in more recent years, including detailed analysis of Y chromosome lineages once mistakenly associated with a Khazar or Slavic origin. Early, lower-resolution genetic studies had noted that a lineage common among Ashkenazi Levites belonged to a broad haplogroup, also common, across Eastern Europe. And some researchers floated the possibility that this reflected a Slavic or Khazar man who joined an early Ashkenazi community and became the ancestor of a large share of later Levites. Higher resolution sequencing, published in 2017, revisited that same lineage with far greater genetic precision and traced its specific branch back instead to a Middle Eastern paternal ancestor who likely lived around 1,700 years ago, correcting the earlier, lower-resolution assumption and substantially weakening the case for a large-scale Khazar contribution to Ashkenazi ancestry specifically. None of these individual findings, however, could be fully anchored to a confirmed starting point because none of them had actual ancient Israelite genetic material to compare against. Every conclusion was an inference, built from the present, reaching backward into a past nobody could directly sample. The Kiryat Ye'arim discovery changed that, even if only partially and only as a first step. The results, presented at an academic conference in October 2023 and published in more detail in the scholarly volume New Studies in the Archaeology of Jerusalem, focused first on the genetic material easiest to recover and interpret reliably, the Y chromosome, passed down exclusively through the paternal line, and mitochondrial DNA, passed down exclusively through the maternal line, both of which tend to survive in degraded ancient samples more readily than the full genome, and can be sequenced with a higher degree of confidence from smaller amounts of recovered material. The male individual from the tomb carried a Y chromosome lineage belonging to haplogroup J2, a paternal lineage broadly distributed across the modern Middle East today, and found at meaningful frequencies among a range of Jewish and non-Jewish populations across the region. A lineage with deep roots in the ancient Near East that long predates the Iron Age burial itself. Mitochondrial DNA recovered from the burial identified haplogroups T1A and H87, lineages whose broader distribution patterns point toward ancestral connections reaching into Anatolia and the Caucasus, alongside components more typically associated with the southern Near East and Arabian Peninsula, a combination that fits comfortably within what researchers already understood about the wider ancestral makeup of the region without offering any dramatic surprise of its own. That research, led by geneticist Mark Haber and colleagues, analyzed ancient DNA recovered from 93 individuals excavated at sites across the southern Levant, including Tel Megiddo, Tel Hazor, and Tel Abel Beth Maacah in northern Israel, a site called Yehud in central Israel, and Baca in central Jordan, combined with 20 previously published ancient genomes from the wider region. These sites span a wide geographic and chronological range, giving researchers a far broader picture than any single excavation could offer on its own, letting them track how ancestry shifted, or in many cases did not shift, across different city-states and different centuries within the same general region. After filtering out closely related individuals to avoid skewing the results, a standard precaution in population genetics that prevents a handful of close relatives within one family from being mistaken for a broader population trend, researchers compared the remaining ancient samples against a reference database of more than 1,600 modern individuals from populations across the region and beyond. The conclusion was striking. Present-day Jewish communities and Arabic-speaking populations native to the region were both found to derive at least half of their genetic ancestry from the Bronze Age Canaanite population that inhabited the southern Levant roughly 3,500 to 1,200 years before the Common Era, the same broad cultural and demographic world described in the Hebrew Bible as existing alongside, and eventually giving way to, the emerging identity of ancient Israel. The study also found that Canaanite populations across separate city-states scattered throughout the region, despite the political fragmentation typical of the Bronze Age southern Levant, with each city-state often functioning as its own independent political entity, remained genetically cohesive with one another across a span of roughly 1,500 years, more closely related to each other than to distant populations, suggesting a shared regional population that persisted through many generations of local political change before Israelite identity itself emerged from within or alongside that same broader Canaanite world. The researchers additionally identified evidence of later population movement into the region from the direction of the Zagros Mountains and the Caucasus, sometime between roughly 2,500 and 1,000 BCE. An additional ancestral layer that, together with the older Canaanite base, contributes to the genetic makeup of nearly every present-day population native to the southern Levant. a group that includes modern Jewish communities, Palestinian and other Arabic-speaking populations, and additional regional groups including Druze and Samaritan communities, all of whom carry some combination of these same deep ancestral layers in different proportions, shaped by which later migrations, conversions, and periods of relative isolation each specific community experienced over the centuries that followed. Taken together, these two lines of evidence, the broad population-level Canaanite ancestry study from 2020 and the first direct sampling of confirmed First Temple period remains in 2023, do something neither could fully accomplish alone. The 2020 study established at a population level that the deep genetic roots beneath both ancient Israel and its Canaanite neighbors were shared and regionally continuous, rather than the product of some later population entirely displacing an earlier one, a finding built from dozens of individuals spread across many sites and many centuries, giving it real statistical weight even without a single sample dated precisely to the biblical kingdoms themselves. The 2023 discovery, though still partial, finally gave researchers an actual reference point from inside that world, a real genetic sample from a real family that lived and died in Judah during the biblical First Temple period, against which every later theory about where Jewish communities came from and how their ancestry changed over 2,000 years of dispersal, conversion, and intermarriage can now begin to be tested directly rather than only inferred. Neither study on its own closes the case. Together they mark the first time the field has had both a wide, population-level picture and a specific dated point of direct contact with the world that picture describes, two different kinds of evidence finally converging on the same historical window, rather than approaching it separately from a distance. It is worth being precise about what this evidence does and does not establish, because this is a subject where imprecision spreads quickly and unhelpfully, and where the temptation to draw bigger conclusions than the data actually supports is a real and recurring problem in how findings like these get discussed once they leave the pages of an academic journal. The genetic findings do not settle questions of religious identity, legal status under Jewish law, or personal connection to a specific ancestral community, none of which are questions genetics is equipped to answer, and no serious researcher in this field has ever claimed otherwise. Jewish identity, both historically and today, has always been shaped by religious law, community, conversion, marriage, and lived practice, not by any single genetic marker or ancestry percentage, and researchers working in this field have been consistently careful to say so, often more careful than the headlines summarizing their work. What the evidence does establish is considerably narrower and still significant. It confirms that a shared, regionally rooted ancestry connects ancient Canaanite populations, the biblical kingdoms of Israel and Judah, and a wide range of present-day populations native to the southern Levant, Jewish and non-Jewish alike, in proportions that differ by community, but draw from the same deep ancestral wells rather than from entirely separate, unrelated sources. It provides, for the first time, an actual physical genetic sample from inside the First Temple period itself, rather than a reconstruction built entirely from modern populations working backward through assumption and inference. And it offers a direct evidence-based correction to at least one popular alternative theory of Jewish origin, showing that the genetic case for a large-scale Khazar origin, once treated as a serious possibility in both academic and popular discussion for the better part of a century, does not hold up against higher-resolution paternal lineage data pointing instead toward a Middle Eastern founding ancestor for one of its most cited pieces of supporting evidence. What remains genuinely unresolved is also worth stating clearly, and there is more of it than a single headline finding might suggest. The full autosomal genome data from the two Kiryat Ye'arim individuals, the roughly 90% of the genome that would allow far more detailed comparison against both ancient and modern reference populations, had not yet been fully published at the time these preliminary Y chromosome and mitochondrial results were shared, and researchers explicitly described more complete findings as still to come, likely to include far more granular detail about exactly how these two individuals compare against other ancient Near Eastern populations, the Bronze Age Canaanite samples from the 2020 study, and modern populations living in the region and beyond today. With only 2 individuals sampled from a single family tomb, researchers are careful not to treat these results as representative of the entire Judahite population of the era. Any more than 2 people sampled from a single modern family could be assumed to represent an entire nation, and future excavations recovering additional First Temple period burials from other sites across the region will be needed before researchers can speak with real confidence about broader population-level patterns rather than the genetic profile of one specific family. Exactly how the ancestry visible in this Iron Age tomb connects, generation by generation, to the ancestry found in later Second Temple period Judeans, and from there through nearly 2,000 years of diaspora to the distinct modern Jewish communities of today, including Ashkenazi, Sephardi, and Mizrahi populations that each carry their own additional layers of regional admixture accumulated across the centuries in Europe, the Iberian Peninsula, and various parts of the Middle East and North Africa, respectively, remains an active and ongoing area of research rather than a fully mapped chain of evidence. Each of those transitions, biblical Judah to Second Temple Judea, Second Temple Judea, to the early diaspora communities and the early diaspora to the modern world, still needs its own direct ancient evidence, its own excavated burials, and its own successfully sequenced genomes before the full 2,000-year picture can be considered complete rather than partially inferred. There is something worth sitting with in how long it actually took to reach even this partial answer. Modern genetics as a field has existed for well over a century, since the rediscovery of Mendelian inheritance at the start of the 20th century, and the double helix structure of DNA itself has been understood since 1953. Genetic studies of Jewish populations specifically have been conducted since at least the early 20th century, using whatever tools were available at each stage, from blood type surveys in the early decades to modern whole genome sequencing today. And DNA-based ancestry research has been a serious, active field for more than 3 decades, producing hundreds of papers on Jewish population genetics alone. And yet the very first confirmed ancient DNA sample from a person who could be called an Israelite or Judahite in the most direct, unambiguous sense, someone who lived and died inside the world, the entire question was actually about, was not recovered until 2023. Buried the entire time in a modest family tomb on a hillside a short drive from Jerusalem, protected, ironically, by the same looting and disturbance that had once threatened to destroy it, since whatever the original looters were searching for clearly was not human bone. And they left behind exactly the evidence later generations would need most. Every theory proposed before that point, however carefully researched, however widely cited in academic literature or popular writing, was built without ever being able to check its answer directly against the source. That is not a criticism of the researchers who came before, many of whom did careful, honest work with the only tools and samples available to them at the time. It is simply a reflection of how genuinely difficult ancient DNA preservation is in this particular climate and this particular span of time, and how much patience a question like this one actually demands before science can finally offer something more solid than an educated guess built from thousands of years downstream. What makes this discovery worth remembering is not that it resolves every question people have long asked about Jewish origins, because it clearly does not, and researchers involved have been careful never to claim otherwise. It is that after 2,000 years of dispersal After centuries of scholarly argument built entirely from the outside looking in, science finally has a real physical starting point to work from, an actual family buried in Judah during the era of the First Temple, whose bones have finally had something to say. For as long as this question has existed, every answer offered was, in a very real sense, a story told about people nobody could actually reach, reconstructed from fragments scattered across 2,000 years and half a dozen continents. Now, for the first time, there is a fixed point at the beginning of that story, one small family tomb that survived, largely by accident, everything that happened to the land around it in the centuries since. The rest of the picture, the full 2,000 years connecting that family tomb to the diverse Jewish communities living across the world today, is still being assembled one careful excavation and one painstaking DNA extraction at a time, likely to be filled in gradually over the coming years as more Iron Age and Second Temple period burials are located, sampled, and sequenced, using the same patient techniques that finally worked at Kiryat Ye'arim. Each new burial that yields usable DNA adds another fixed point to a timeline that, until very recently, had none at all, slowly turning a chain of inference stretching back 2,000 years into something closer to a chain of actual evidence. This case also fits into a broader pattern showing up across ancient DNA research more generally. Over the past decade and a half, this particular discovery illustrates with unusual clarity. Time and again, questions that scholars had spent generations arguing over using only indirect evidence, comparative linguistics, archaeology, and the study of modern populations working backward, have finally started being tested directly once ancient DNA extraction techniques advanced enough to recover usable genetic material from increasingly degraded, increasingly ancient remains. The technical breakthrough that made the Kiryat Ye'arim result possible, targeting the petrous bone rather than more commonly sampled skeletal material, was itself only refined and widely adopted within roughly the last decade, meaning a discovery like this one was, in a very real sense, technically impossible. For most of the time, this particular question has been debated. What changed was not the burial itself, undisturbed and waiting in that hillside cave for roughly 27 centuries. What changed was the field finally catching up to it, developing tools sensitive enough to read what had been sitting there, intact and legible, in a strictly biochemical sense, the entire time. If a single family tomb on a quiet hillside outside Jerusalem was enough to finally anchor 2,000 years of scholarly debate to real physical evidence, what other ancient mysteries are still waiting on one specific burial, sitting undisturbed, for researchers to finally find it? Let me know which ancient population story you would want scientists to test with direct ancient DNA next in the comments below. And if you want to see what the fuller Kiryat Ye'arim genome reveals once it's published, and where this research goes from here, make sure to like this video, subscribe to the channel, and turn on notifications so you don't miss what they find next.